Okay, so welcome to this next video on at the activation of T cells. Okay, so we got to the stage where we had basically activated our nuclear factor of activated T cells. We had removed those phosphate groups from the serine-rich region here, and now we had exposed this nuclear import domain here. And that nuclear import domain was going to result in the nuclear factor of activated T cells being translocated into the nucleus, and now we will denote it the NFAT-N to denote that it's present in the nucleus. So this has now gone in to the nucleus of the cell, and basically what it is it's a very potent transcription factor. Okay, it alone is not a transcription factor, so maybe I should call it a transcriptional coactivator instead. And basically what it does is it associates with CFOS and CJUN transcription factors. So where should I put these? So if we've got a gene here, let's firstly draw our gene. Okay, here. Then the gene, let's say the gene is here. So this is our gene. And basically, upstream of all genes, you have what's known as a promoter region. So this is the promoter region. And the promoter region is where certain proteins combined in order to influence the expression of the gene. So this is the promoter region. Now, the nuclear factor of activated T cells is in some cases a transcriptional factor, a transcription factor on its own accord, but often it's the case that it also works alongside other transcription factors. So there are two very famous transcription factors known as CFOS and CJUN. So I'll put these here. So let's say this one is CFOS and this one here is CJUN. And basically, CFOS and CJUN form heterodimers together, basically. So you get a CJUN with a CFOS, and they together are very, very potent transcription factors. Basically, what the nuclear factor of activated T cells does is it binds to these CFOS-CJUN heterodimers and forms a trimer, basically. So this is an NFAT now in the nucleus, so I'll denote that by the little n at the end, okay? And it binds with these CFOS-CJUN dimers, and now this complex is going to affect transcription in a different way to the way that the CFOS and CJUN heterodimers affected transcription. So it's going to bind to different promoter regions and have different effects. And basically, what it does is it binds to promoter regions of certain genes and it increases their expression. And two genes that are really important, which it increases the expression of, are the interleukin-2 gene. So interleukin-2 gene. So it increases the expression of this interleukin-2 gene, and therefore you get more interleukin-2 being made. And it also increases the expression of the interleukin-2 receptor alpha protein. So interleukin-2 receptor alpha component of the interleukin-2 receptor. Okay, now that's quite a mouthful. So instead, often interleukin-2 is denoted IL-2, and the interleukin-2 receptor alpha component is denoted the IL-2R alpha. Okay, right. Now, what is interleukin-2 and interleukin-2 receptor alpha going to do? So overall, let's just summarize where we've got to so far. The activation of signal 1 and signal 2 within the T cell has now resulted in an increased production of interleukin-2 and interleukin-2 receptor alpha. Yes, we've seen all the intermediate steps, but this is the overall effect that you've now had, basically. You've increased your production of interleukin-2 and you've increased your production of the interleukin-2 receptor alpha. Now, basically, what happens is the interleukin-2 receptor alpha goes to the membrane of the T cell. So let's say this is the T cell membrane here, okay? Basically, the interleukin receptor on the surface of the T cell membrane is a trimer. It's made up out of 3 pieces. So let's say this is one of the pieces here, this is another one, and this is the final one over here. Right. Now, in normal T cells that are not activated, you have two of the components of the interleukin-2 receptor. You have the interleukin-2 receptor beta component, and you have the interleukin-2 receptor gamma component. But you do not have the interleukin-2 receptor alpha component, and you cannot have a fully functional interleukin-2 receptor until you have all three components. So I'll repeat that. In normal quiescent T cells which have not been activated by an antigen-presenting cell, you do not have the alpha component of the interleukin-2 receptor. You have the interleukin-2 receptor beta component, and you have the interleukin-2 receptor gamma component, but not the alpha component. So, firstly, when you activate your T cell by stimulating it with an antigen-presenting cell, it's going to start producing, finally, this interleukin-2 receptor alpha. Also, what's going to happen is that it's going to produce the ligand for this receptor. It's going to produce interleukin-2. So now it's got the full receptor for interleukin-2 here, and it's also got the ligand for the interleukin-2 receptor. So what's going to happen is interleukin-2 is going to be released by the cell, and it's going to basically take part in autostimulation. It's going to go back onto the cell that released it and stimulate that cell's interleukin-2 receptor, which is now newly formed. Okay? And now what happens is that this interleukin-2 receptor causes the T cell to divide. It activates growth stimulatory pathways. And there are many of these growth stimulatory pathways which lead the cell to divide. Growth stimulatory pathways. Okay, and what that overall leads to, as I say, is the T cell proliferating. So you copy the T cell many, many times. T cell divides. So overall, what has happened basically is that when the antigen-presenting cell came and activated this T cell by giving it signal 1 and signal 2, then that has caused the T cell to overall start proliferating, which makes sense because this T cell has the correct receptor on its surface that will bind to this antigen that the antigen-presenting cell is presenting it. Now, this stimulation here by the interleukin-2 receptor of the T cell to divide is then known as signal 3, and it's the final signal that you need to get in order to get T cell activation. And T cell activation really means this T cell division. So T cell division is basically T cell activation. That's what we mean when we say the T cell is activated. It means that the T cell is dividing, basically. T cell activation. Right. Okay, so one last drug that I want to talk about is an antibody against the interleukin-2 receptor. So there is an antibody which we have made which can bind to the interleukin-2 receptor and therefore stop interleukin-2 from being able to bind to the interleukin-2 receptor. And this antibody is known as basiliximab. Basiliximab, they always have fantastic names, monoclonal antibodies. So basiliximab is a monoclonal antibody, which means that it's a single antibody that will bind to a single epitope of the interleukin-2 receptor. And basically, it comes, binds to this extracellular domain, stops interleukin-2 from being able to bind, and therefore you stop signal 3. You stop this final signal of T cell activation, and therefore you don't stimulate the growth of the T cell. So again, basiliximab is an extremely potent immunosuppressive agent.